Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're continuing our lesson today on meiosis. Today I really just wanted to focus on some important vocabulary and some important things that are happening during meiosis. So the stuff that we're talking about today, this is all happening in meiosis. It's all very important to making those gametes correctly, and I just kind of wanted to review some vocabulary with y'all so that this stuff isn't tripping you up in the future. So I'm going to go over some of these terms again with y'all, and then I've got a little review worksheet that we'll practice on when we're done. So make sure you're following along so that that worksheet is going to give you no problems later on. So let's go ahead and hop into it and see what's going on. Okay, so I wanted to talk about prophase 1. There's a couple of important things that are happening in prophase 1. First of all, let's talk about the word synapsis. This is a verb, synapsis. What is happening during this process? This is when homologous chromosomes pair up to form a tetrad. What is a tetrad? Well, remember that prefix tetra means 4, and so there's 4 sister chromatids all kind of lined up in a row. Okay, there's going to be one chromosome next to another chromosome. Those together is called a tetrad. Okay, and that's because there's 4 sister chromatids all lined up. When that tetrad is formed, they're going to swap some genetic material. That exchanging of genetic material is called crossing over. When crossing over happens, they exchange their genetic material between those non-sister chromatids of the homologous chromosome. Okay? This process is going to increase genetic variation by creating new combinations of alleles. So again, this crossing over, this is why you and your siblings don't all look just alike, because you each got a little bit different information from your parents. Okay? So these events, the synapsis, the homologous chromosomes, and the crossing over, these events are all critical for genetic diversity and do not occur during prophase of mitosis. So everything I just talked about, this is exclusive to meiosis and does not happen in mitosis. Okay, here's some other words. So that tetrad, remember that a tetrad is formed when the synapse happens. Okay, homologous chromosomes come together and align to form a tetrad consisting of 4 sister chromatids. Remember that 2 homologous chromosomes, and they each have 2 sister chromatids each. So that's a total of 4. This process is crucial for ensuring that each resulting sex cell ends up with only half the genetic material, contributing to the reduction in chromosome number and promoting genetic diversity. So this process is very, very important to meiosis occurring correctly. Okay, and then let's just take another look at that crossing over. What is crossing over? Well, it's when the chromosomes are paired as a tetrad. So this right here, if you can kind of get a look at this picture, this is a tetrad. We have 2 chromosomes right next to each other. Okay, each chromosome has 2 sister chromatids. So like one side of this chromosome is a sister chromatid. They come together. Coming together is known as synapsis. When synapsis happens, it forms a tetrad. This is called a tetrad. When the tetrad is formed, crossing over happens. When crossing over happens, they switch their genetic material. So you can see we started with all blue and all purple, but through this process of crossing over, we're ending with this one has a little bit of purple and this one has a little bit of blue because they've swapped some genetic material. Okay, so those kind of things all happen very close together. They're all in prophase. Okay, first you make the homologous chromosomes come together. These are homologous chromosomes. The homologous chromosomes form a tetrad. This is called synapsis. When the homologous chromosomes form a tetrad, they cross over. Okay, so all of that is happening in prophase 1. Okay. Crossing over, this process involves the exchange of segments of genetic material between homologous chromosomes during the formation of tetrads. It results in new combinations of genes and contributes to genetic diversity in offspring. Okay, this process introduces genetic variation by exchanging segments of genetic material between the homologous chromosomes. This variation is a key factor in the diversity of genetic information passed to offspring and can drive evolutionary changes. This is where a lot of evolution is kind of coming from, because we're mixing up those genes so much and we're making individuals that are different from each other on purpose. We're doing this intentionally to increase genetic diversity. Okay, great. So all of that happens in prophase 1. Let's talk about something really important that happens in anaphase 1. Okay, during anaphase 1 of meiosis, those tetrads separate as homologous chromosomes as they're pulled apart, and the pairs of chromatids move towards opposite poles of the cell. But that doesn't always happen the way that they're supposed to. Sometimes those chromosomes do not get pulled apart. When that happens, it's known as non-disjunction, okay? Non-disjunction means they're not separating the way that they're supposed to. And what this means is that the resulting cells are going to have the wrong number of chromosomes. They might have an extra chromosome or missing a chromosome, and that's going to lead to a lot of genetic problems down the road, okay? So let's take a look at that non-disjunction in a little bit more detail. So again, non-disjunction, this error can occur in either anaphase 1 or anaphase 2 of meiosis, but it is happening in anaphase. In both cases, it leads to the improper separation of chromosomes, which results in gametes with an abnormal number of chromosomes. As a result of non-disjunction, some gametes may end up with 24 chromosomes, while others only have 22. So again, what's happening here? Well, let's kind of take a look at what's going on. So here we have 46 chromosomes. That's the number that you want. But during meiosis, if they don't all separate correctly, if non-disjunction happens, then we might get cells with the wrong number of chromosomes. So this one got one extra and this one got one fewer than it was supposed to. That means that this cell has 47 chromosomes and this one only has 45. Well, if non-disjunction happens again, we're going to get cells that the gametes are going to have the wrong number of chromosomes. Now this one has 24 up top, this one has 22 at the bottom. These two are fine, but if either one of these are used in that fertilization process, then the zygote and the ultimate organism that's made as a result of it will have too few or too many chromosomes, and that's going to have a lot of problems down the road. And this is all a result of non-disjunction, which is when the chromosomes don't separate properly, okay? So when that happens, you can see here that if we use those cells as gametes and they do fertilize, you can see what will happen here. Well, 22 chromosomes from this cell, 23 from this cell. This zygote will only have 45 total, and that means most likely this individual, this zygote, will die. It's missing information. It doesn't have all the information it needs to live. So if you're missing chromosomes, that's really, really bad. You're not going to survive. Okay. On the other end, a little bit different. Here, these gametes, this one has one extra chromosome. When they fertilize, now the zygote will have an extra one. It has 47 instead of 46. This zygote will maybe survive, but if it does, it will have some birth defects. Okay, so getting the correct number of chromosomes is very critical to your ultimate survival. The most common type in this situation, when you get an extra chromosome, the most common type is called Down syndrome, which we'll talk about in a little bit more detail later on. But people with Down syndrome, that is a result of just having one extra chromosome, okay? Great, y'all. Thank y'all so much for following along with me. That was great. I've got some review stuff I want to do with y'all. So teacher, if you could, why don't you real quick pass those out, pass that worksheet out to everybody, and when you're ready, come back to me and I will review it with y'all, okay? All right, I know that didn't give y'all any problem, but just in case it did, I'm going to review it with y'all just to make sure we're all on the same page. So y'all feel free to change or add anything that you don't have. So let's see what everybody came up with. All right, so number one says 2 chromosomes that have the same genes at the same location but may have different alleles. So what would we call that? These 2 chromosomes, they're similar size, similar shape, they have roughly the same genetic material, but they might be different forms of it. So you might remember that those are called homologous chromosomes. Okay, so let's go with letter B for that one. Okay, let's move on. Number 2. A cell that contains only one set of chromosomes with only one member of each chromosome pair. All right, so for this one, what do you call it when it only has one set of chromosomes? It doesn't have the full 46 that it's supposed to, it only has 23. What kind of cell is that? Well, if it has half the number of a regular cell, that would be a haploid cell, which is letter A. Okay, A is haploid, so let's use that one. All right, let's keep it going. So number 3 here says, a cell that contains 2 sets of chromosomes, including both members of each homologous chromosome pair. So now what do we call it when you have the full 46 chromosomes that most of your cells do? Not haploid, but instead diploid, which is letter O. So let's go with that one, okay? Great work, let's keep it going. So 4 says, a type of cell division that results in 2 genetically identical diploid daughter cells, each with the same number of chromosomes as the original cell. So this is describing either mitosis or meiosis. Which one do y'all think it's describing? Well, I see 2 genetically identical diploid cells. I know that that's going to be mitosis, right? So for number 4, let's go with mitosis, which is letter D. Great work. All right, number 5, the stage of meiosis where homologous chromosomes pairs are separated and moved to opposite poles of the cell. Okay, so I've kind of mentioned this a couple times, but I'm going to go ahead and mention it now again. When I see this word, homologous chromosomes, okay, that tells me that I have to be in meiosis 1. Okay, because we're really only dealing with homologous chromosomes in meiosis 1. By the time you get to meiosis 2, there aren't any homologous chromosomes left because they've all been separated. Okay, so now I know whatever answer I pick is going to have a 1 after it. So now all I have to do is figure out which part of PMAT is it. So this says the stage of meiosis where the homologous chromosomes are paired up and separate and move to opposite sides of the cell. So what do we call it when they're pulled apart from each other? What's that called? So being pulled apart is known as anaphase, and because it says homologous chromosomes, we know that it's anaphase 1, okay? So anaphase 1 for 5, and that's going to be letter C. So just remember that. If you see homologous chromosomes, it's one of the meiosis 1, okay? If you see the words sister chromatids, that's letting you know that it's meiosis 2, okay? So let's go to 6 here. 6 says, the stage of meiosis where chromosomes, each consisting of 2 sister chromatids, line up at the cell's equilateral plane. Okay, so let's see. A stage where the chromosomes, each consisting of 2 sister chromatids, line up in the cell. So I don't see anything about homologous chromosomes, it just says chromosomes, and then it says 2 sister chromatids. So to me, I just told y'all if it has the word sister chromatids, it's probably going to be meiosis 2. And so this is going to be meiosis 2. We just have to figure out what part of PMAT lines up in the middle. Which one lines up in the middle? That's metaphase, right? And because it's talking about sister chromatids, it's going to be metaphase 2. So 6 is metaphase 2, which is going to be letter M, okay? M for metaphase. All right, let's keep it going. 7, the stage of meiosis when tetrads, homologous chromosome pairs, form and crossing over happens. Okay, again, so I see homologous chromosomes, so that lets me know that I'm in meiosis 1, okay? So now I just have to figure out which part of the PMAT is it. Well, I know when it says the tetrads form and crossing over occurs, remember that all happens in prophase. Prophase is the longest, it's the most complex part of meiosis and mitosis. So if you're not sure, you can usually just guess prophase because that's when most of the stuff happens. Okay, so this is going to be prophase 1 for 7, and that is letter N. So let's go with N for that one. Okay, great work. Let's keep this going. All right, I'll scroll down a little bit here. Okay, 8 says, the stage of meiosis where homologous chromosome pairs line up along the middle of the cell. So you can see why these keywords, picking out these keywords is very important because it's hard to tell if it's meiosis 1 or meiosis 2. But using these keywords, you can kind of figure it out. This says homologous chromosomes. So I know it has to be meiosis 1 because that's the only time that we have homologous chromosomes. So what is it called when they line up in the middle of the cell? What's that called? Well, I know that that's metaphase, and because it says homologous chromosomes, I know it's metaphase 1, okay? So letter 8, for letter 8, let's go with E, which is metaphase 1. Okay, 9 says, the stage of meiosis where sister chromatids are separated and move towards opposite poles of the cell. Again, I see the word sister chromatid, so that tells me that I'm in meiosis 2, and then I just have to figure out move towards opposite poles of the cell. Well, what does that describe? Moving apart is anaphase, and because it says sister chromatids, I know it's anaphase 2, okay? So for number 9, let's go with letter L, which is anaphase 2. Okay, great work. Let's keep this going. So 10, this says the division of meiosis that reduces the chromosome number by half, resulting in haploid cells. Okay, the division of meiosis that reduces the chromosome number by half, resulting in 2 haploid cells. All right, so for number 10, I have F. Okay, and F is meiosis 1. Okay, so meiosis 1, we start off as diploid cells. Through meiosis 1, we've cut the number of chromosomes in half by the end of it. Okay, let's keep it going. 11, the division of meiosis that results in the separation of sister chromatids producing single-stranded chromosomes. Okay, so the division of meiosis that results, I see sister chromatids again. Okay, so that's letting me know that I'm in meiosis 2, and this one says division producing single-stranded chromosomes. All right, so for 11, I have K, and that's meiosis 2. So we did meiosis 1 and then meiosis 2 down here. Okay, so just a couple more. 12. 12 says the stage at the end of meiosis 1 where 2 cells are formed, each with double-stranded chromosomes and half the number of chromosomes. Well, really, because this says the stage at the end of meiosis 1, we know that the last part of PMAT is telophase, and because this says the end of meiosis 1, we know it's probably telophase 1. All right, so for 12, let's go with letter G. Okay. Y'all are doing great, just a couple more. 13 says the stage at the end of meiosis 2 where 4 haploid cells are produced. Same thing, the last part of PMAT is telophase, and because this is meiosis 2, it's going to be telophase 2. So for 13, let's go with J, telophase 2. All right, couple more. 14, a structure formed during meiosis consisting of a pair of homologous chromosomes, each made up of 2 sister chromatids. So what is it called when I have those 2 chromosomes right next to each other? What's that called again? Each of these is a chromosome. Each chromosome has 2 sister chromatids. So I have a total of 4 sister chromatids here. That's called a tetrad. Okay, so for 14, let's go with letter H is tetrad. All right, last one, 15. The process during prophase 1 of meiosis where homologous chromosomes exchange genetic material leading to genetic variation. Again, we know what it is at this point because of process of elimination, but I do see homologous chromosomes, and just as a reminder, if you see homologous chromosomes, it tells you you're in meiosis 1, and this whole process is describing prophase. Okay, so for 15, I have I, which is crossing over. Okay, so crossing over for that last one. That's when you exchange genetic material, right? All right, so great work. That stuff is tricky. I just wanted to make sure I reviewed some of those important vocabulary words with you. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.